Hi everyone, thanks for watching this presentation. My name is Dave Madge and I'm here to tell you a little about the Biotech platform for discovery of bifunctional molecules, and in particular molecules that work by targeted protein degradation. After a little background information, I'll go on to talk about how we support drug discovery in this space, and finally say a few words about the next steps in the development of our toolbox. Whilst the majority of the chemistry and biology I will describe has been applied in the field of PROTAC drug discovery, many of these tools are equally applicable to other forms of bifunctional molecules, and in particular, any that seek to manage the interactions of two or more proteins. I'll discuss some of the chemistry, biophysical methods, structural biology, computational chemistry, and functional assays. In fact, the breadth of application of what we now know as bifunctional molecules turns out to be quite diverse. Of recent significant interest are those molecules that bring housekeeping proteins into contact with target proteins whose levels we wish to reduce, a strategy of targeted protein degradation. But beyond this application, we find molecules that interfere with protein-protein interactions, molecules that redirect the immune system, stabilize protein complexes, and modulate biological interactions. Some of these molecules are complex, sometimes cyclic species with more than one protein interacting phase, including some natural products. Other molecules are constructed by the linking of multiple ligands that each bind to different proteins. It's that latter model that we will focus on today, illustrated on this slide by the degrader DBET1, which comprises a ligand for the target protein BRD4, coupled through a short linker to a ligand for the housekeeping ligase cereblon. Within the preclinical discovery teams at Biotech, we have built an end-to-end platform for discovery of therapeutic small molecules, as well as peptides and oligonucleotides. Our philosophy is to develop capabilities across all aspects of drug discovery for all of the different therapeutic strategies we support, including bifunctional molecules. This slide provides a very high-level overview of those capabilities, but on the next few slides, I'll dive into some of these areas in more detail. Specifically, I'd like to spend a little time discussing ligand identification and optimization, ADME, biophysics, structural biology, and functional assays, both in vitro and in vivo. Let's start by talking about how we might go about finding ligands for the proteins we might want to interact with. For example, members of the E3 ligase family, or for the target proteins we wish to bind to simultaneously. I mentioned on this slide 4 of the most widely used hit-finding technologies, and these are all applied in our labs. 2 platforms in particular lend themselves to hit-finding in this area, and these are the 2 affinity selection methods at the top of this screen. Affinity selection, mass spectrometry, and screening of DNA-encoded libraries. These 2 techniques are agnostic of binding site on the target protein, and also identify ligands that do not necessarily have functional effect. Since the functional effect on the target protein is not a requirement for targeted degradation strategies, this approach allows us to scan more of the protein surface for suitable interaction sites. This is especially powerful when coupled with access to large, diverse screening collections, allowing high coverage of chemical space. DNA-encoded libraries are a perfect way to explore as much chemical space as possible, and we have been building DELs for some time, amassing more than 200 libraries and around 80 billion molecules, each encoded by a DNA tag and available for affinity screening. These libraries, applied to a selection process with several cycles, with identification of bound ligands by PCR and next-generation sequencing, have proven invaluable for rapid ligand finding for diverse targets. Mass spectrometry-based screening applications have also evolved rapidly in recent years, and we have added affinity selection by mass spec to our toolbox. This platform enables a plate-based assessment of protein binding for hundreds of thousands of small molecules quickly and efficiently. With this technique, there is no need for protein immobilization, and assay development is typically very rapid. We've been applying this technique to our screening collection of 300,000 small molecules to very good effect across multiple target classes. With ligands for the protein of interest and the housekeeping protein in hand, the next task is to build these into molecules that allow for simultaneous binding of both moieties to their cognate receptors. We've been working in this space for 5 years now and have a chemistry team of over 600 supporting more than 50 clients developing bifunctional molecules. This team works closely with our purification and analysis platform, itself typically responsible for processing more than 500,000 molecules each year, as well as with our ADME, biophysics, and biology teams, whose work I will discuss in a few slides' time. The chemistry in this area is increasingly diverse, with ligands for a widening collection of ligases becoming available. Inventive linking chemistries play a role in determining physical chemical properties, metabolism, and permeability of bifunctional molecules. As part of this chemistry effort, we have a growing collection of preformed ligand plus linker chemistries available for rapid development of proof of concept degrader molecules. Large-scale synthesis of PROTAC molecules is also supporting our teams, including multi-kilo scale synthesis. Such complex molecules present specific challenges for purification and analysis. Solubility is often poor, providing headaches for HPLC analysis and purification. Stability may also be a problem, especially in the presence of regular HPLC modifiers. And multi-step purification strategies are often required. However, systematic method development and the processing of thousands of samples with hundreds of methods and column types now allows us to rapidly identify appropriate purification and analysis protocols, incorporating the use of optimized organic modifiers. We now routinely purify tens of thousands of bifunctional molecules each year. Many of these complications apply equally to in vitro and in vivo pharmacokinetic analysis of PROTAC molecules. We've been developing and optimizing analytical methods in DMPK for these molecules for several years now, and routinely work on several thousand such molecules every year, with extensive application of triple quad mass spectrometry, as well as UPLC to separate and quantify these molecules in many different biological matrices with high precision. In order to demonstrate desirable protein interactions, we've built capabilities to support production of high-quality recombinant proteins and platforms to characterize protein-ligand interactions and complex formation. We make extensive use of biophysical methods such as SPR and MST, as well as employing protein crystallography to precisely visualize interactions, providing information for further optimization. Biophysical methods are especially important, allowing us to determine the kinetics of interaction of a bifunctional molecule with both of its cognate targets individually, as well as investigating the formation of the ternary complex required for degrader function. This latter complex is the species whose formation we are especially interested in. In fact, with these methods, we can identify the cooperative nature with which the bifunctional molecule enhances formation of the ternary complex with the ligases and the target protein. I can show you a little data for this on the next slide. If we just focus on the two plots on the right-hand side of this slide, we can see that the interactions promoted between VHL and target protein B at the top are stronger and longer lasting compared to those promoted with target protein A below. As you can imagine, we can equally keep the target proteins constant and examine the effect of different degrader molecules. Furthermore, we can quantify the degree to which affinity of the degrader molecule in the binary complex increases when we move to the ternary complex. On this slide, we show data for the ligand CC885, which shows around a 40-fold increase in affinity for the ternary complex versus the binary complex. In other words, a very strong positive cooperativity. Of course, we can also visualize these ternary complexes by X-ray. And when we do so, the information can be transformational with respect to enabling rational ligand optimization for one or both of the binding sites that bifunctional molecules engage with. These structural insights can be used in other ways, and recent examples suggest that using this information to design cyclic analogs of the degrader ligand can lead to molecules with similar functional activity, perhaps enhanced stability, permeability, or penetration deep into dense tissue beds such as tumors. Let's move downstream now and examine some tools to measure the in vitro activity of bifunctional molecules. On the next few slides, I'll describe some assay platforms that can characterize interactions with the protein of interest, with a target ligase, as well as formation of the ternary complex. We'll also show methods to quantify the desired functional outcome of protein degradation. A useful technique for investigating binding to the target protein, especially where a tracer ligand for the desired binding site is available or can be prepared, is the TR-FRET assay technology, illustrated here for a kinase target. After careful setup and optimization, this can be invaluable for rapid selection amongst a pool of candidate warhead ligands for the protein of interest. A further useful platform is the nanobret system, applied here to investigate the cellular permeability of candidate degraders, as well as their engagement with the ligase VHL. This provides a whole cell target engagement assay to aid selection for more complex downstream investigations. Complementing the biophysical techniques mentioned earlier, we can also visualize ternary complex formation using the alpha-LISA technology, illustrated here for the ligand-induced complexation of the ligase VHL with the bromodomain BRD4, facilitated by the PROTAC molecule MZ1. Use of high-quality reagents and careful optimization provides a very sensitive assay system for quantifying ternary complex formation. Furthermore, to demonstrate in-cell target degradation, we've implemented a variation on the HiBit platform that allows for a dual endpoint analysis of both the degree of protein degradation as well as the kinetics of that degradation. This is enabled through antibiotic control of the LG bit status of different groups of cells. With LG bit off, in the absence of doxorubicin, we have an endpoint assay. But in the presence of doxorubicin, LG bit is on, and we are able to run a kinetic assay. The HiBit platform can also be used in primary cells, illustrated here with an application in primary immune cells. In this example, we're looking at the degradation of STAT3 in CD3 positive cells, induced by a cerebron-binding PROTAC molecule. This sort of assay setup allows us to visualize further functional endpoints, and I'll show you some of that data on the next slide. Here we've looked at the effect of a degrader molecule at 3 separate endpoints in parallel. This is work in primary immune cells, and we are visualizing protein knockdown by around 1 order of magnitude, as shown in the left panel, as well as visualizing rescue from immune cell-triggered suppression of T-cell proliferation in the middle panel, and effects on levels of the cytokine interferon gamma in the right-hand panel. In order to quickly confirm functional effects of new protein degraders, we can also run quantitative immunofluorescence studies, sometimes known as in-cell Western studies. In this protocol, we would first optimize seeding density and ensure we have a good assay window with healthy cells and good DMSO tolerance. And then we can generate a reliable quantitative measure of degradation of target protein with very good reproducibility. Of course, many of the initial investigations of the application of degrader molecules have been in the oncology therapeutic area. Hence, it's important to be able to demonstrate in vivo effects on tumors as a marker of efficacy. In this experiment, we demonstrate a dose proportional reduction in tumor volume and tumor weight in new mice implanted with GP5D tumor cells. Looking to the future, it's clear that we will extend the collection of E3 ligases that PROTAC molecules target as a means of achieving localization of protein degradation in specific cell types or tissue beds, as well as perhaps driving enhanced selectivity for the target protein. We are actively working to further populate this space with additional proteins, ligands, and assays to enable a much broader mining of the opportunity this modality provides. We also see increasing applications of computational selection and design tools. And I've been optimizing a workflow exemplified here that couples linker and ligand conformation sampling, as well as examining the protein-protein interface enabled by the bifunctional molecules. This approach provides a means of making informed selections for both ligands and linkers prior to synthesis. I'm sure we'll see many further developments like these, as well as many more molecules entering development and confirming the clinical utility of hijacking this pathway. Please do contact me if you would like to discuss our capabilities in this area in more detail. And thank you very much for your attention.